proceed with population ecology. Okay, why populations change in size? Kenapa population boleh berubah, become smaller, become bigger? Okay, the population change in size due to 5 factors. Either due to biotic factors, death rates, birth rates, immigration or emigrations, and also abiotic factors. Okay, how to estimate the population size? There are two ways to estimate population size. One is quadrat sampling technique. This quadrat sampling technique for the plant. Another one is capture, mark, release, and recapture technique for freely moving organisms such as animals and humans, mostly for animals lah. Okay, quadrat sampling technique is used to estimate the number and also distributions of organisms that do not move, so mostly refer to plants. And we use quadrats that will be placed randomly to represent plants in our habitats. More quadrats being used will increase accuracy of data. So here is the example of quadrats being used for quadrat sampling technique. And by using these quadrats, kita akan count the number of each plant dalam quadrat ini. And bear in mind, awak kena make sure berapa area of the quadrat being used. Sebab nanti kita nak calculate. Okay, there are 3 formulas to be memorized for quadrat sampling technique. Ada 3 formula yang awak kena ingat. Okay, the first one is percentage frequency. Okay, percentage frequency is used to estimate the number of times the species can be found when a quadrat is thrown. Bila awak campak quadrat tu, berapa banyak kalikah awak boleh jumpa spesies itu? That is percentage frequency. So here is the formula. Percentage frequency is the number of quadrats containing species divided by the total number of quadrat sample times with 100. Okay, second formula is the density of species that is used to estimate the existence of plants population. Formula for density of species is total number of individual of species in all quadrats divided by number of quadrat samples times with area of each quadrat sample. Okay. The third formula is the percentage coverage that is used to estimate the area of quadrat that occupied by a species. For the percentage coverage, the formula is the area covered by all quadrats in meter square divided by number of total quadrat samples times with the area of quadrats and times with hundreds. So, okay, let's see the first question. Okay, kita tengok contoh yang pertama. Okay, you can use your calculator. Okay, for the first question, this is the data obtained when our students use quadrat sampling techniques to find the percentage covered by species of grass pea in the school field. The area used is 1 meter square. So here the result. The question asks the percentage coverage. So the formula for the percentage coverage is the area cover of all quadrats in meter square divided by number of quadrat samples times with the area of quadrats area times with 100. So how to calculate? Okay, area covered by all quadrats. Okay, there are 6 quadrats. 1, 2, 3, 4, 5, 6. 6 quadrats. And all of the 6 quadrats have grass pea. Semua quadrats ini ada grass pea. So area covered by all quadrats Awak just total up 0.35 plus 0.75 plus 0.68 plus 0.14 plus 0.25, okay, plus 0.10, and then you divide with number of quadrat samples. Total number of quadrats, we have 6 quadrat samples, and the area of quadrat area is 1 meter square. So we times with 100. So we get answer D. Easy? Okay, next example. Okay, table 2 shows the result of an experiment to estimate the number of plant P in 10 quadrats with area, okay, area 1 meter times with 1 meter. So this is the area of the quadrat. Okay, total number of quadrat is 10, okay, but not all quadrats occupied by species P. Okay, so for the percentage frequency, the question asks you to calculate the frequency of plant P. Okay, so percentage frequency is the number of quadrat containing species divided by total number of quadrat sample times with 100. So we identify the quadrat that contains species. Quadrat 2, quadrat 4, quadrat 5, quadrat 6, quadrat 8, and quadrat 9. So number of quadrat containing species. 1, 2, 3, 4, 5, 6. 6 quadrat containing species divide by total number of quadrats being used 10 times with hundreds. So we get C. Easy? Easy, right? Okay. Okay, for capture, mark, release, and recapture technique is used to estimate the populations of animals that move freely. Okay, the first one we capture for the first time, we randomly capture, and then we mark that animals either using ring or tags or colors, but this mark make sure are not attractive to the predators. Jangan terlampau bright in color, jangan terlampau obvious sampai boleh attract predators. And these marks are not poisonous and not limit the movements of these animals. And this one is censored, so I will detect. And then we will release these animals. Kita bebaskan these animals. And then at the certain times kita akan recapture for the second time. Kita tangkap balik. And then we record the number of the marked organism. Kita kira yang kita mark yang first time tadi. Okay, here is the formula to calculate the population size for freely moving animals. Okay, number of organisms in the first sample times with the number of organisms in the second sample. Okay, total number of first sample, total number for the second sample, divide by number of marked organism recaptured. Bahagikan dengan organisms yang ada mark sahaja. Waktu second times kita recaptured.